Thank you very much. Introduction. First of all, I'd like to express the congratulations for the 30th anniversary of ACC. It's very much honor for me to be opportunity to give the presentation here. So I'd like to show the title like current status and prospects of culture collection in microbiology. So summarize, I'd like to express the importance of the taxonomy for culture collection today. So I visited KACC first in 2005, 20 years ago. It was 10th anniversary of KACC. As Marsan said, 20 years ago, so many of us are very young, not only myself, Dr. Hon is also very young. So I enjoyed the Korean food there at the home of Dr. Go, at the head of this KACC. So today's content, first, I introduce the influence of the reclassification based on the genomics in prokaryote taxonomy with two examples. And second, the usefulness of correct identification for the use of the microbial resources in culture collections. First, now is the time for the transition from 16S RNA sequence to the whole genome genomics. So there's some confusion in the taxonomy, but many information is coming, as Dr. Ma said, from 16S to the whole genome. And then the next second one is that many of the microbiologists are not so much interested in taxonomy because they think the taxonomy is not so informative for their science research. But it is not true, then I'd like to introduce this one. So generally, this I already introduced, like from 16S now to the whole genome sequence is now becoming popular. Then they are used for the taxonomy. And then the procedure of the identification or taxonomic study is now become clear. So many microbiologists are now involved in the taxonomy because the name are given by the sequencing. So simple. Formerly, only the experts can make experiments for the taxonomy. But now, as a result of such kind of a change of the situations, many single strain species have proposed. So many species are now. So even I'm studying taxonomy for more than 30 years, but I cannot imagine from the name so many species or genus names are present. And but that is not so much, not good for the author to propose a new taxa. Don't characterize enough for the new taxa. On the way, the new information is coming like a flood. So in culture collection, many microorganisms are received by the culture collection, and before preservation, they need the quality management for the preservation. The most important thing is the identity between the strain from the depositor and the received ones. Then the 16S RNA sequence is very convenient. Only one technique for all the microorganisms, all the prokaryotes can be checked. And at the same time, the 16S RNA is very important for the proposal of the new taxa. Then the author, to propose the new taxa, have to deposit 3 things. One is submit the paper, and at the same time, or before, the sequence data like 16S and genome are also registered. And then the culture collection must accept the deposit of the possible type strain candidates, and culture collection have to issue the certificate for the submission of the journal. it's very, very hard work for the culture collection because only the culture collection is a place to handle the materials. The first example is the transition from 16S RNA gene-based phylogeny to whole genome-based phylogenomics. As you know, the lactobacillus is a very popular name of the bacteria, lactic acid bacteria, and then it was divided into 23 genera. And then now, the Lactobacillusaceae has 26 genera. This is very big work for the taxonomy because Lactobacillus plantarum is now Lactopyrantibacillus plantarum. The Lactobacillus casei is now lactic casei Bacillus casei. So then all the they are divided into many genera and change their genus names like this. But these division can be done based on the core genome tree from the phylogenomics. But many of the genera are based on their species classification. Then many of us are very much comprehensive for corresponding between the species and the new genus. Because the Lactobacillus is a very, very large genus, and then it is natural to divide into so small. And the most important thing is for this one is a correlation between the classification based on the 16S and the full genome analysis. This shows the good correlation between the whole genome classification and the 16S classification. The phylogenetic tree is based on 16S, and the genus division is done by the whole genome analysis, but they are good correlation, and then you can see the genus classification only by the 16S and go to the second step. This is the latter half. So the usefulness of 16S RNA, first it can be used for the first stage of identification. And the metagenomic analysis based on the 16S is also useful to determine their genus names. And culture collection essentially can rename the strains only based on the 16S RNA. For example, the genus name, such as Lactobacillus sp, is changed to the divided into Fructolactobacillus sp or something like this. Because most of the culture collection has their own 16S RNA sequence data, even not for non-type strains. Then the next example is the case for the family Erythrobacteriaceae. This is a very important group of the proteobacteria. And this is also done in the whole genome analysis in 2020, like this. This family was composed of 7 genera. And they are good correlations are shown in the genus classification and the 16S RNA classification. Then they can identify their genus by 16S. Then the new classification is done by whole genome analysis using a phylogenetic tree based on the 288 single copy ortholog protein. Then the problem is occurred like the former 7 genera are now present here, and the red genera are added to this family. And in the phylogenomic tree shows the corresponding to the genus like this. And if some genus are divided into the several genera, there is no problem. But the problem is, for example, former Erythrobacter is divided into many, and the classification is based on the position of the type species for the genus name. Then many of the other species are separated from the type strain in phylogenomic tree. Then, for example, the genus Oranticibacter contains these kind of the former named Erythrobacter. And where is the Erythrobacter? Type strain is here. Then Erythrobacter, genus Erythrobacter contains mostly Porphyrobacterium, formerly named Porphyrobacterium, is good for the one genus based on the phylogenome. Then it means this means the genera are mixed in the base 16S RNA phylogenetic tree. So this means in the new classification, we cannot give the genus names only by the 16S RNA sequences. So one genus is divided into several places by 16S RNA sequences. So culture collection have not only the type strains, many strains, isolated strain, non-type strains have the name given by, for example, the 16S RNA like in the previous tree. But now only the 16S RNA cannot give the name for the genus, even the genus. It also affects for the metagenomic analysis based on the only 16S RNA. They can give the analysis as a result with the genus name. So to summarize this, the recipient of the strains from the culture collection must check the updated names of the strains from the database. This means formally, in the old day catalog, they give the name at least 16S. But after that, even after received, the name may have changed. Then the author have to, recipient have to check the current name by catalog of the collection or other publications. The collection also must catch up the taxonomy, and sometimes culture collection must carry out additional experiments for the identification. This means just when we find the name changed in the journal, it's easy to change the name by culture collection, but non-type strain, we need the additional experiments for the give the genus names. Whole genome sequence is now becoming essential identification mandatory for the proposal of the new species in IGSEM. But the identification based on the 16S RNA sequence may be re-evaluated, how to use the 16S RNA sequence. If we set this is a kind of the rule to give the genus name, it may be convenient. But it does not match with the whole genome analysis phylogenetic tree. Now it is a problem in the transition age. So this is one example, such kind of the difference. And the second example is gaps between LPSN and the valid publication in the IGSEM. The LPSN is a database of the scientific names. And many of the microbiologists using bacteria, they check the names from LPSN is so popular for the non-taxonomists. But we have some problem between the gap between the LPSN and valid publication in IGSEM. Many of the people believe LPSN is the database for the IGSM publication, but not always so. Then now I introduce this one example. You know, genus Plevotera. Plevotera is an anaerobic gram-negative bacteria. It's very important for the analysis of the intestinal microbiomes. In the study on 2022, the big genus Plevotera divided into several new genera, like Segatera, Hoysera, Leirera, Parentia. Then it is divided into such kind of a genus. It means the 77 species of the genus Plevotera from the 77, 25 species, 5 species are separated from the genus Plevotera and to propose transfer to the new genera like this. And then the Plevotera populi is now transferred to the Segatera in the paper, and the Plevotera populi is very important species for the intestinal microbiomes. And only one new genus is proposed for the new species. Then you understand all the 25 species are transferred from the previous Plevotera, and they are a kind of the new combination names. And this paper is out of the IGSM, but it is found in the validation list in the IGSM, and this is these 12 species of the genus Segatera are now validly published. LPSN is, as mentioned, this is a very important database many microbiologists use. That is now maintained in the DSMZ by Dr. Mark Gawker. But just after publication of that new proposal of the new genera, the LPSN does not accept the change of the names and retained them as the Plevotera. Then, as a result, the genus Segatera is a synonym of the Plevotera. It means the separation of the Segatera is not accepted by LPSN, even they are published validly in IGSEM. Then the other genera, like these genera, at the same time separated from Plevotera with the Segatera, are also treated as the synonym. Then in LPSN, we search the Segatera copri, then that is a synonym, and the name, correct name is Plevotera copri is the previous name, kept in the previous names. Then we search the Plevotera copri, that is the correct name. In such situation, the genus Segatera is not accepted by LPSN, but validly published. In such situation, there is a proposal of the new species of the genus Segatera. It is done by my colleague in the Tokyo University of Agriculture, and the name is Segatera asaccharophila. Then that is IGSEM and validly published and now appearing in the LPSN. But it is found as a Segatera asaccharophila, but it is noted as the taxonomic status is orphaned. Taxonomic status is orphaned species. What is orphaned species? Orphaned species is a species belong to the not accepted genus. It means Segatera is not accepted by LPSN, then many Segatera species should be used as previous name Plevotera. But this is a new species of the Segatera. Then there is no name for the Plevotera asaccharophila. Then, in the list of the species of the Segatera, many of the 12 species of Segatera are synonym of the Plevotera, but only this one, Segatera asaccharophila, is orphaned species. Then it means LPSN does not give the correct name to this species. It has the difference between the LPSN and IGSM valid publication. Then many non-taxonomists believe or give the priority to LPSN. But we taxonomists should follow the IGSM valid publication. There is a gap or the dilemma. Then now I want to show the responsible culture collection to keep the type strains and such classification gaps between LPSN and IGSEM. So this is the catalog of the KCTC. KCTC is kindly accept the new proposal of the new species, like Plevotera copri is now they use the Segatera copri. The classification new Segatera is accepted. In such situation, the Segatera copri is and the Plevotera copri is the synonym. The first choice is Segatera. JCM also, Segatera is a priority, has a priority. And DSMZ also, it is very interesting because the Segatera copri has the first choice and no name of the Plevotera copri anymore, like this. And then Segatera, we search in Segatera copri, that is found in Segatera copri. And in DSMZ has no choice of the synonym. Only just Segatera copri is shown and no more Plevotera copri here. But it is interesting because DSMZ is a host institute of LPSN, like this. The promotion of the LPSN is next to the Segatera. It's very interesting. The two have a different idea. Then the new species, Segatera asaccharophila, is found in KCTC catalog like this, because Segatera asaccharophila is validly published, and only LPSN called the orphan name, but LPSN and KCTC proposed show the Segatera name. This is the same as JCM also. And DSM also, they all have naturally Segatera asaccharophila, the new species, is shown here. Unfortunately, these species are not in the NBRC and KACC, but all the collection can do the same thing to follow the valid publication in the JCM. Many of the microbiologists maybe have the LPSN is correct, but the culture collection, all the culture collection follow the IGSEM, and it is, I understand the networking of the culture collection is so important against LPSN. So this is a summary. And genus Segatera, Hoysera, and such kind of the separation is accepted by the IGSEM, and for culture collection follow such kind of the change. And then the culture collection welcome the new species of the Segatera. So the DSM, GCM, KCTC accept the Segatera and don't follow LPSN. The culture collection is, as I mentioned, as the materials are handled, then we have to follow the depositor and then give the name. So then it means the culture, it's very important for culture collections to work together. The third one is the usefulness of the correct identification and promotion of the use of micro resources in culture collections. So actinomyces is very important for antibiotic producer. So many such microbiologists search a new organism to produce new materials. Then, in old days, many small number of the characteristics like morphology or physiology, they propose the names or identified. And then now, Streptomyces has almost 1,000 species. Then it is very difficult to find a partner for the DNA-DNA hybridization in long time. Then so much confusion. Confusion means the genetically same species, but with a different name, or vice versa. The typical example that microbiologists are not interested in the taxonomy. It means many of you know Streptomyces cerevisiae A32 is a very popular actinomycete for the study using the genetic study. Then the Streptomyces cerevisiae A32 is not the type strain, then not representing this species. And then later, the taxonomist identified the strain and found this is not the cerevisiae, and this strain is Arabidoflavus. And proposed that this change is the name, and the culture collection follow this change. Then cerevisiae, and then different way, the cerevisiae is the type strain of the cerevisiae is identified as cerevisiae A32 is now re-identified as Violaceo ruba. On the other side, the type strain of the cerevisiae was identified as Arabidoflavus. So then, now no more cerevisiae species. But they are still use the A32 with the A32 with the name cerevisiae. But we, culture collection, no more use the name cerevisiae for A32. Then I hope the user of this strain hope to use the Streptomyces Violaceo ruba A32. But many paper accept the paper using the cerevisiae A32. Then the microbiologist using actinomycetes for the such kind of the development of the new antibiotics. Now, for the genomic age, they can find the synthesis genes of the polypeptide synthase or non-ribosomal protein synthase are good target for the searching the antibiotic producers. This is a synthetic pathway of the enzyme complex. Very large enzyme complex with a cluster are present in such antibiotic producers. But that it does not follow the taxonomy like this. The many antibiotic producers using the name the Streptomyces hygroscopics. It is a very popular species for the antibiotic producers. But recently, classification based on the genomics changed the names like these. Streptomyces hygroscopicus to these kind of the different species. Different species show different products. the product gene so large, then not so many genes are equipped in the species. Now they already changed the names, but still these are Streptomyces hygroscopix, but lower similarity to the type strain still remained, and they have the good producer of such kind of antibiotics. Then many of the microbiologists believe the production of the antibiotics is strain dependent and not related to taxonomy. It is not good for the taxonomist. Then in the study of the antibiotics producer, this is a result study of the Dr. Komaki of the NBRC. The strict meaning of the hygroscopics is here, and there are some producers still here, and they have the name hygroscopics. But other producers also have the many interesting species, but separated from the hygroscopics with the separate names. And some are not still in the hygroscopics name, but they should be changed names. At the same time, Streptomyces albus is also the antibiotic producers, and it is in the same situation. It is also still interesting, and some are regarded as a synonym. Some different species are synonym of albus, or some albus are separated from the type strain. Then now reclassification is ongoing based on the 16th and whole genome analysis. And the whole genome analysis study found the gene location of the gene clusters are interesting and correlation to the taxonomy. Like this is the Streptomyces albus species in the different strain, shows a similar layer of the gene clusters. But the species, separate strains separated from albus has the different gene clusters. So it means the taxonomy and correlation of the gene cluster of the polypeptide sequence are very good. So taxonomy may be, in summarize, the taxonomy will be more useful for the efficient screening or reduce the amount of the international transportation, such kind of things, so efficient use. So,
Sensei, I am sorry. You have 3 minutes more, Sensei. I'm sorry.
Okay.
Yeah.
Time?
Yeah. Time is up.
Okay. Then culture collection should provide the material with such kind of the suitable name. But for the type strain is very easy, but the non-type strain is very, very important for the correct identification, but it should be done by the culture collection. Okay. Then this is the culture collection needs. This is my talk, and how important for the culture collection to be the good taxonomy. And so first I visited the ACC in 2005 as the 10th anniversary. But after that, in 2009, I visited here the ACC in Suwon. And this, so Dr. Hong is still young and still young. And then at the meeting, we started the collaboration with KACC and NBRC. Then after that, KACC has many, many interesting isolates, and we play a part of the taxonomic characterization. And since then, the first paper is coming out in the 2011 with the name Dr. Kwon, the former head of the KACC, and my name. And main work is Dr. Tamura, Hamada, Dr. Weon, or others. And since then, more than 38 publications until the 2024. Now generation changed. I disappeared, but Hamada and other your important persons are working together. It's very, very important for the collaboration with the culture collection in the playing separate roles. Then, actually, 38 publications with the new 12 new genera, 48 new species. So that is so large contribution for the enrichment of the culture collections. So mainly, actinomycetes we are interested in, then these species are proposed in the collaboration. New species, new genera, and they are, so many are deposited in KACC and NBRC. So the final conclusion, and the culture collection are responsible for the materials to supply. Collections must catch up the updated information and technique for the community. And important to us to establish a network of culture collections, share them in ACM and abroad. So finally, thanks to Dr. Komaki, Hamada, and Tamura for the assistance of the preparation of the slide. Thank you for attention.